Welcome to Epithelial Tissues Part 2. What we're going to get into right away is how epithelial tissue is categorized, talk about how the function of epithelial tissue correlates to some of its structural characteristics, and then we'll talk about the structure and function of selected epithelial tissues. So let's start first by talking about how epithelial tissue is categorized. One way that it's categorized is on the number of cell layers. A simple epithelium is a single layer of cells that sit over an underlying tissue. Right? Stratified epithelium has two or more layers of cells sitting on top of an underlying tissue. It could be as few as two, or you could have many layers of cells overlying a tissue. The second way that epithelial tissue is categorized is based on the shape of the cell. Some cells are said to be squamous, right? They're thin and flattened. So a thin, flat cell might look like that. I know it's kind of lopsided, but you get the picture. A cuboidal cell is cube-shaped. In other words, it is about as wide as it is tall as it is deep. And then the third common cell shape is columnar. And columnar cells are tall and thin, or tall and narrow. And so in the naming of an epithelial tissue, the name will typically include the word simple or the word stratified to indicate the number of layers, and the term squamous, cuboidal, or columnar to indicate the shape of some of the cells. Now, the cell shape or the cell size actually has a functional consequence to it, right? The size of the cell relates to how much cytoplasm it's going to have. So when you look at squamous cells, right? Squamous cells are thin and flattened, whereas cuboidal cells are about as wide as they are tall, whereas columnar cells are narrow and tall, right? So you notice squamous cells, because of their size, are not going to have much volume or space on the inside, whereas cuboidal cells are going to have even more space on the inside. Or if we look at this in more simplified terms, if you think of the amount of blue shading, right, in each of these simplistic diagrams as being equivalent or proportional to the amount of cytoplasm, what we can see is that squamous cells have the least cytoplasm and columnar cells have the most. Now, the reason why this is significant is because most of a cell's activity occurs in the cytoplasm. That's where proteins and other molecules are made. It's where ATP is made. It's where wastes and other molecules are broken down and recycled. So the amount of cytoplasm a cell has is directly related to the amount of work or activity that the cell can perform. And so squamous cells are the least active and columnar cells are the most active, right? And this is going to relate to what these tissues or cells can do. Let's look at another aspect of epithelial tissue structure, right? Now, one thing epithelial tissue does is it acts as a barrier, or in other words, it's a lining that separates spaces. And sometimes these linings are designed to allow the easy and rapid movement of materials from one space across the epithelial tissue to another space. And other times the lining is designed to be to reduce or make it difficult for something to pass through the epithelial tissue to the other side. Let's take a look at this in the context of diffusion. Remember, diffusion is the movement of a substance from an area of high concentration to an area of low concentration, and it doesn't involve the cell being active. It's simply powered by molecular movement inherent in the molecule. You can see here some hypothetical times that it would take the average molecule to move a certain distance. And at this distance, right, 1/1000th of a micron, it takes 0.0005 milliseconds for something to diffuse. That's so fast, you can't even comprehend how fast that is. Right? If in order for that same molecule to diffuse a 100th of a micrometer, it would take 0.05 of a millisecond. To go 0.1 of a micrometer, it takes 5 seconds. To go 1 millimeter, it takes 8.5 minutes. Well, some of our cells start to die after 8.5 minutes without any oxygen, right? And so the idea here is that relatively small increases in distance dramatically increase the amount of time it takes for something to move. So if you look at this situation here, oops, I'm sorry. Yeah, well, we're just going to go with this. We're going to do this in one take. So when you look at this situation right here, where you have a single layer of thin cells, it would be really easy for this red molecule to diffuse through it because the distance is small, and so it's going to diffuse rapidly. Here, the distance between the apical surface and the basal surface is longer, and so it's going to take this red molecule longer to diffuse because it's a greater distance. Here, it takes even longer to get through because the distance from the top of the columnar cell to the basal surface of the columnar cell is larger, right? And over here, I have a stratified tissue. Each one of these squares or rectangles represents a cell. I have many, many cells stacked one on top of the other, making a very thick layer. It would take a super long time for that molecule to diffuse through. And so what this boils down to is that simple epithelia, right, whether it's columnar or squamous, is thin enough to be easily passed through. All of these are relatively permeable, but they're not particularly protective because it's only a single layer. This is the most permeable. This is slightly less permeable. Slightly less permeable still. But these stratified tissues, they're really hard to get through, so they're not very permeable, but that makes them really good at protecting. A stratified epithelium is going to be more protective and serve as more of a barrier that prevents things from moving, which is why we see, for example, the urinary bladder lined with a stratified epithelium so that urine doesn't leak out into the other parts of the body, right? Or that the skin and the tongue and the inside of your mouth are lined with this because it's subject to abrasion. I'm going to go ahead and stop here so we can go ahead and talk about all of the specific epithelial tissues in one video.